let's say that I said, pick one process of ATP and energy transfer and explain what's happening. Okay? Let's just say that I said that, Maria, okay? One example, two example, three example, four example. Okay? Four examples right here of ATP and how it is transferring energy to other processes. Other processes making disaccharide, making polysaccharide, making protein. Those are the other processes. When we say transfer the energy, we're transferring it on to other things in the body. All right? This one here shows that when ATP loses the third phosphate through the process of hydrolysis, energy is released that was stored in this bond. That energy can be used for such cellular processes as active transport, muscle contractions, or the movement of flagellum on the male gamete. As a result of that reaction, the products of that reaction are ADP and phosphate, which is given off inorganically. Those can be recombined through the process of phosphorylation or rephosphorylation, if you want to get technical, back into ATP. That's one example.
So what is the catabolic reaction part?
Catabolic reaction?
Yeah, like it's on the arrow with the rephosphorylation.
Catabolic, remember, anabolic and catabolic are both types of metabolic reactions. Anabolic builds, so the rephosphorylation would be an anabolic reaction. The energized glucose here that goes on to make the disaccharide would be an anabolic. This energized glucose that goes on to making starch would be an anabolic. Or the protein. All of those really could be anabolic reactions. Catabolic is tear down. So the only thing we're tearing down is this ATP molecule. Okay, we're tearing it down in order to break it up and do something with all of the products of it. All right, that's what we were just looking at, the anabolic, catabolic part of that. Where's the term not over here? Is it over here?
Yeah, like where it says
right here, rephosphorylation, catabolic reactions, rephosphorylation. Okay. So they're just looking at in the terms of adding one phosphate to an ADP or adding two phosphates to an AMP. Okay, it's a build and tear down and a build and tear down. That whole ATP cycle is a build and tear down and a build and tear down. That's what they're just referencing it all, that they all could be catabolic reactions or anabolic reactions, depending on which way you want to go with it. That's one example. Another way that ATP transfers energy in our body is to take the ATP molecule, break the third phosphate away, and glucose, which we would have brought into the cell because we fuel our body with this primary source of energy called glucose. It's glucose already passed through the membrane by facilitated diffusion. It's now in our cytoplasm. And ATP this time, when it breaks away the third phosphate, that phosphate joins to a glucose molecule, and when it does that, glucose becomes energized. Right? Once it becomes energized, so it built catabolically. Here you're looking at it stealing the energy, so it released it, and then it built. Energizes the glucose. At that point, glucose doesn't need the phosphate anymore, so it breaks it off. It's given off inorganically. And that energized glucose goes on to making a disaccharide. Okay, so it can be like glucose and fructose, combining them together. Next one, again, we're going to break the third phosphate. This time, we're going to follow the ADP molecule. It's like this one up here follows the phosphate, this one follows the ADP, because it's one molecule. Remember, when we break it, we get two products. We get that, we get that. So this process here is kind of addressing what can happen to the phosphate. This one's addressing what can become of the ADP. It's still beneficial. Remember, it's second string. Okay? Your second string player is still as valuable as the first. Okay? So it's looking at this point of that this one is hooking up to the glucose, energizes the glucose by that temporary hookup, doesn't need it anymore, so it'll break off. The ADP is given off. This energized glucose then goes on to making starch, which is glucose to glucose to glucose to glucose. So it's going to need a lot more energy than this one, which only needs to form one bond. And this one needs to form several bonds are going to have to be formed in that creation of a bigger molecule, an anabolic reaction. All right, now the last one is the one that's completely different because this time we're going to break right here. So we're going to get an AMP molecule, and then we're going to get something called a pyrophosphate. In this example, we take the ATP molecule, we break the bond between phosphate number 1 and 2. These two phosphates remain together, and temporarily they are referred to as a pyrophosphate. Then they break, and when they break, it is enough energy along with the initial break, because remember, this isn't the maximum energy. Right here is. So it broke here first, that released a little bit of energy, but not enough. So then we got to break the pyrophosphates. They become inorganic, but that energy combined with the original energy break is enough for an amino acid to hook itself on to the AMP that was left behind, again to energize it, steals the energy, breaks the bond, the AMP is given off to the cell. The energized amino acid goes on to the making of a protein, which is amino bonded to amino bonded, several aminos long. So over here on, in essence, this side of the sheet, but on our right-hand side of our arrow, we're doing it as a chemical equation. We have a bunch of leftovers. We have AMPs, we have inorganic phosphates, we have ADP. Okay, all you got to do is recombine them back, recycle them in a sense, back into another ATP by the process of phosphorylation, which means add phosphate to it. So an ADP would only have to add 1, an AMP would have to add 2, but our body has the ability to do that, plus make new ones every time we bring in glucose. Okay? So those are all examples. It's like one example, second, third. Those are 4 examples, basically. This one actually shows several within one, but those are just examples of how ATP can transfer energy to different cellular processes. We're still inside the cell products. And we're just in that cytoplasm inside the cell, but different means of the way that energy can be utilized. This sheet really is just an information sheet. All this sheet is trying to show you is, first of all, how big, how complex NAD and FAD really are. Okay, they're these huge macromolecules, and basically just showing you that oxidation reduction going hand in hand. NAD and FAD are known to be electron acceptors. That's their jobs. They are to be at the site of a reaction when a molecule is going to be oxidized. This one's going to be oxidized. Here's the glycolysis color sheet. It'll make a little more sense if you really see it for real. Okay, if I use this step right here, like this molecule, see my arrow, so you're looking backwards at what you're used to. Here's your reactant, here's your product. Reactant, product, arrow. Okay, to show the direction. This molecule, from here to here, if I look at the difference in the configuration, I can tell that they are shaped different, so therefore they have to be different. This molecule, I see off to the side, okay, not as part of this molecule, but over here, off to the side, I see NAD, and then an arrow becoming NADH plus H. Right, so NAD I know is an electron acceptor, and it's an electron acceptor in reactions in which oxidation has occurred. As a result, it becomes reduced. So, the only other molecule in this entire reaction, if we're just going from here to here, is this molecule right here. So I know this molecule had to have been oxidized because NAD was reduced to NADH. So basically, like on this coloring sheet, this molecule here, if you look at this one, that is what they're trying to represent with this oval shape right here. This molecule loses 2 hydrogen atoms. So that's what it's telling me that's going right here. This molecule is losing 2 hydrogen, and from that hydrogen, then the NAD will take the electrons in 1 proton. So that's why we write it as NADH plus H, because that extra H, okay, doesn't attach in the same location. And again, explained on this sheet, as they show you, these all up here, but this one down here by itself. It doesn't attach when it's attached. And then we get a new molecule formed over here, okay, compared to this one. So when you look at your numbers, okay, and you could do each and every one with your counts of your carbon to hydrogens to oxygens, but the names are a little easier sometimes than trying to just do your chemical formulas for each one of those. But NAD is reduced to NADH because that's NAD's job. It's an electron acceptor. This is oxidized, NAD is reduced. That's all this sheet is really just trying to show you, is that oxidation and reduction are reactions that occur at the same time. And if something's going to be oxidized, NAD or FAD have to be there as the bystander to take on those electrons. Okay? They're going to come back into play in the end. These NADHs that are formed here, they have value. They still do have value. Okay, we got to keep track of them as we do each one of these sheets. And we'll see when we get to the very last sheet, all of these are going to be recycled back into NAD so that there's a constant supply. If there's not a constant supply of NAD right here, this whole thing shuts down. So there has to be a recycling process that goes on within this whole cellular respiration system. Remember the big picture, okay, the cellular respiration, C6, H12O6, in the presence of oxygen, okay, so glucose in the presence of oxygen, because it's aerobic, reacts together. We're going to get 6 waters, 6 carbon dioxide, you exhale carbon dioxide, plus energy. And that is the formula, chemical formula for cellular respiration. Glucose, primary fuel for our cells. In the presence of oxygen, this is aerobic respiration. You've got to have oxygen. It will be the final electron acceptor. Right now, NAD and FAD are accepting the electrons. When we get to the end, oxygen is going to be our final electron acceptor. That's why it's needed. As a result, we're going to make waters when this happens. We're going to make carbon dioxides, and we're after energy. We're after ATP. We're going to get 36 of them per glucose. Okay. What's unique about this formula, this chemical reaction here, is you take what's over here and you move it on this side. You take this and move it over there. You now have the chemical equation for photosynthesis. So that's the strong relationship between the animal and the plant processes and why we need each other. It's not as simple as just, we exhale carbon dioxide, they take our carbon dioxide, and they give off oxygen back to us. In this, there's a relationship with these two. They're going to be inverts of each other. The products of one is the reactants of the other. The only difference is energy. We're after a chemical energy. And you need to build these ATP molecules. Photosynthesis is going to be light energy and sun to drive it. We just change our energy conversion, but it's the same. You learn it once, you know 2 formulas. You just have to put it on the side. Right? So, but the big thing here is this arrow means so much. It's 4 of these sheets.